The topic of my presentation is documentation and reporting of investigational protocol. So today we will learn about objective of these points. Before to start my topic, I will give a brief introduction about the clinical research. Clinical research is the comprehensive study of the safety and effectiveness of the most promising advances in the patient care. There are two types of clinical research, observational and clinical trial. As according to my topic, I'll focus more about on clinical trial. Clinical trials study the safety and the effectiveness of intervention and procedure on people's health. The goal is to find better ways of preventive diagnosis and treating diseases. They are used to test drugs and medication, new types of surgery, medical devices, and in ways of using current treatment, new ways of changing health behavior, new ways to improve the quality of life for sick patients. So why clinical research is important in the HSCT? It is very important to know about the history and about the clinical research. So there is a famous saying, only he or she knows the past has a future. Of course, we should know the history of why this clinical research is very, very important in HSCT. HSCT was evolved from the field that was declared dead in 1960 after explosion of atomic bomb in World War II. The survivor of immediate exposure died due to bone marrow failure. Researchers were directed to treat the lethal condition, led to discovery that bone marrow derived stem cell from healthy donor could replace the hematopoiesis after total body radiation, and TBI can be used to eradicate the aberrant hematopoiesis. In order to improve the outcome, the believers joined forces. They met each other, openly reviewed their cases and charts one by one, exchanged views on hurdle and opportunities, spent time together on the slope in Delft, and became friendly rivals of EBMT. So, how this EBMT was born? And with the passage of time, the HSCT rapidly diversified in the term of donor types, stem cell sources, indications, management of the complication, and HSCT become open to centers with no links to radiobiology and no longer bound to sterile units and to selected the countries. Informal gathering and database no longer sufficient to share their urgently needed information exchange, and EBMT become a formal structure and become open to all involved in the patient care and scientific analysis. So on the basis of past experiments, this global network become active, and the world organization involved in the HSCT such as the WBMT, EBMT, AFIBMT, CBIMTR. CBMTR is actually the part of EBMT. The EBMT database now holds information on more than 500,000 transplants. Over 35,000 new patients were treated annually over the last 5 years. And HSCT was saved 400,000 lives, but there are stories of many transplants with no clear outcome must not be repeated. There are no data about the future. From an empirical scientific point of view, it is impossible to say what lies ahead of us, according to the Spire. So, on the experiment of our past and current situation, we can decide and plan our future. For HSCTs to remain, valuable treatment, mentalities, and methods of the past no longer sufficient. The idea of beliefs, hence physician creating their own Goldilocks condition that are the more favorable condition that led to the end of HSCT. It has to replace by the stringent scientific approach. WHO guiding principle for cell, organs, and tissue transplant, data collection, and data analysis are integral part of the therapy. Need to become a mandatory reality for transplant team. Reporting has to become a real time and lifelong to close gap between transplant numbers and reporting. Data and quality management should be condition sine qua non, absolutely necessary for all with close interaction between local, national, and international organization. Outcome of transplanted patients need to correct comparison with non-transplant strategy on routine basis. I'll give you example. Suppose we are doing transplant of this thalassemia patient, and we should must comparison with non-treating HSCT, and they are on the other any other treatment, alternative treatment like gene therapy on or any other treatment for thalassemia. It must be reported, and we should be comparison for outcome. So good clinical research practice. Good clinical research practice is a process that incorporates establish ethical and scientific quality standard for the design, conducting, reporting, and recording of clinical research involving the participation of the human subjects. There are basic principles for clinical research. Researcher involved human should be scientifically sound and conducted in the accordance with the basic ethical principles, which based on the declaration of the Helsinki. And it should be scientifically justified and a clear protocol, foreseeable risk and discomfort, anticipated benefits, and initiated with only if the anticipated benefit for the individual's researcher, subject, and society clearly outweigh the risk, and receive the independent ethics committee, institutions review IBR approval for the prior to initiation, and conducting in the compliance with the protocol, approved protocol. Freely given informed consent should be continued if the only benefit risk profile the remain favorable. Qualified and duly licensed medical personnel, physician, or when appropriate, should be responsible for the medical care of research subjects and for any medical decision made on their behalf. Each individual involved in conducting a trial should be qualified and educated, training and experience to perform his or her respective task, and currently licensed to do where required. And all clinical trial information should be recorded, handled, and stored in a way that allow its accurate reporting, interpretation, and verification. This is on based on our today's topic. The confidentiality of the records, investigational products in accordance with the applicable good manufacturing practice and should be used in accordance with the approved protocol. Quality of every aseptic of the trial should be implemented. And what steps are to be taken for clinical research? Development of trial investigational protocol, development of standard operating procedure, development of support system and tools, generation approved for the trial related documents, selection of trial sites, and the selection of the properly qualified, trained, and experienced investigator and study personnel. Ethics committee review the approved for the protocol. Reviewing of the regulatory authorities. Clinical trial must be registered with. These are the sites where we can register our clinical studies. UCLN, IC, ISCRTN, BMC, Prespero, and for local Pakistan Health Research Council, Drug Regulatory Authority of Pakistan, Human Organ Transplant Authority. Enrollment of subjects into the study, requirement, eligibility, and informed consent. Consent, the investigational products, quality, handling, and accounting. Trial data acquisition, conducting the trial. And key activities of the clinical trials, safety management and reporting. Where HUTA, that is the Human Organ Transplant, Pakistan Medical Research Council, Drug Regulatory Authority, in that is the local, and for international clinical trial, Central International Blood and Bone Marrow Transplant Research, CBMTR, monitoring the trial, managing trial data, quality assurance of the trial performance and data. Now comes to the investigational protocol, that is our today's topic. The protocol should outline the rationale for study, objective, methodology use, how the data will be managed and analyzed, primary and secondary outcome. It should be highlighted how ethical issues have been considered and where appropriate, how gender issues are being addressed. And these are the, for example, these are the sites where we can register for clinical studies, and where we see this summary page, where these are condition, intervention, location, and study documents can be seen. This is the clinical trial site. And you can see that how we can submit our clinical studies, and how it see the date and the pattern for all this sponsor and information provided by the institute, and group for groups and interventions, outcome and measures, primary and secondary, eligibility criteria. On these all basis, why we do documentation. Why documentation is very important. We should must document in proper way. If it was not documented, it did not happen. So mean that if it's not documented, it means that there nothing work has been done. Purpose of documentation. Documentation for research protocol performed by the clinical program should be maintained in accordance with the institution policy, applicable laws and regulation, including the order approved by the institution or review board, ethics committee, are equivalent correspondence with the regulatory agencies, any adverse event, and the resolution. Types of documents. All records in any form that describes the record, the methods, conduct, and a result of the trial, the factors affecting the trial and the action taken, written informed consent, electronic clinical forms, X-ray scan, and labs. These are all the types of documents. And trial documents are both resource and outcome. These are the resources for regulators and outcome of the study. There are three types of the documentation: trial master file, source documents, and essential documents. So, trial master files should be established at the beginning of trial, both at the investigator institution site at the sponsored file. In the beginning, we must document the baseline documentation and source documents, original documents, data and records should be retained for at least 2 years and then can be destroyed after the written consent for of sponsor. Essential documents that demonstrate the compliance of the investigator, sponsor, monitor with the standard of good clinical practice with all applicable regulatory requirements. They include the investigational brochures, clinical studies protocol, case report, informed consent, and clinical study report. And reporting. Why reporting is very important? On all the base of documentation, we should report accurate, and researchers have the scientific, ethical, and legal obligations to report the result of their research. Review the requirements for reporting results such as the baseline characteristic, progress of the participant through each stage of study, outcome measures, and statistics analysis, adverse events, any specific requirements provided by regulatory monitoring authorities, and ensure the privacy and confidentiality of the study participant whole while providing the supplementary documents record of the study. Some studies specific standard formats are available for reporting, such as gene therapy in the CBMTR. This is the format of CBMTR for reporting. You can see this pattern, how we are reporting on this format. Thanks. Any question?
Thank you. Thank you, Azra. This was a bit complicated topic. And but I